Today I'm very fortunate and very happy to be here to tell you a story of discovery. We as a lab has found a GPCR network in male sensory neurons that promotes the perception of the oviduct. So I will first introduce you to the importance of cell-cell communications and then give you background on what is known on C. elegans active sperm communications. Then I will pop the question of this thesis. Does the environment affect sperm motility? In the results section, I will first provide the evidence showing a HRB, G-protein couple receptor network that functions in males to promote sperm guidance. And then I will characterize the motility abnormality of the SRB mutant sperm. Next, I will show you the SRB endogenous expression patterns and where they maybe function to promote sperm guidance. I will end the result sections with the SRB transcriptional target, focusing on those that could possibly be changing how the sperm is made. First, I would like us to take a step back and appreciate three major inventions that Mother Nature has given us. The very first invention is the invention of life itself in the life form of a prokaryotic cell, a cell without the nucleus. That's a big difference between not living and life. Then Mother Nature continue tweaking this life form and then making a major improvement in the form of a eukaryotic cells. Mother Nature has created a nucleus and other membrane-bound organelles like the mitochondria, and in the process has increased the size of the cells tremendously. As bigger size has more advantage in this world. And then she went on and continued tweaking and make multicellularity, in which case she combined more cells together to form an organism which is more complex and larger in size, which you know that in this world, larger organism will put smaller organism on the menu for breakfast. So it's tremendously advantageous. However, multicellularity has its own requirements, as all the cells in the whole organism must function together harmoniously. So they must be able to communicate to each other. You can easily see example of cell-cell communication just around us. For example, if I have a bruise on my hands, then the damaged cells will release signal and recruit white blood cells to the area to clean up. Another example happened at neuromuscular junctions where the neurons release signal to tell the muscle to contract or relax. And conversely, the muscle also tell the neuron to maintain the synapse, and also in some cases tell the neuron to survive, especially early during neural development. For me, I'm most interested in the communications during fertilization of an egg that's giving positional cue to a sperm to help guide there for fertilization to happen. Normally, observing the sperm motility in the uterus of an animal like a mammal would be technically challenging. Therefore, C. elegans emerged as a very powerful model. As you can see here, through its transparent epidermis, you can see live motor sperm that has been vitally stained moving inside the uterus of a live hermaphrodite. C. elegans is a hermaphroditic species with rare but important occurrence of males. You are looking at a hermaphrodite worm which is gonad stretching from here all the way to here. So the germ cells are dividing and proliferating and maturing and become large oocytes here or here. And then the oocyte then push into this yellow compartment called the spermatheca, where it will fertilize with the sperm, and then the fertilized embryo will be pushed inside into the uterus and slowly being pushed out of the uterus through the vulva. the male sperm travel in the opposite direction. It's enter the uterus through the vulva during the mating process, and then it becomes activated and start moving toward the spermatheca and target here. Using this model system, our lab has previously published 3 papers, one in 2006, one in 2010, and 2013. So basically, we have shown that the oocytes promote sperm guidance by correspondence. Particularly, the oocytes convert polyunsaturated fatty acids from the form of yolk into different F-series correspondents and release them into the uterus. In the uterus, the correspondent form a gradient highest toward the spermatheca, and once the sperm got inside the uterus through the vulva, they become activated. It sends the gradient and target toward the spermatheca. Particularly, I want to emphasize that in the 2013 paper, we have made the following contribution to understanding. We have found that C. elegans oocytes and precursor produce a heterogeneous mixture of more than 10 different types of F-series correspondent that belong to 3 subclasses, F1, F2, and F3. Together, they promote sperm guidance. Also, we have identified the polyunsaturated fatty acid that are the precursor to the F1, F2, and F3 classes. They are the precursor are dihomo gamma linolenic acid, arachidonic acid, omega-3 arachidonic acid, and eicosa pentaenoic acid. Also, we have shown that prostaglandin synthesis in C. elegans hermaphrodites is independent of cyclooxygenase. A year later, we published another paper showing that this process of prostaglandin synthesis that's independent of cauxis is also conserved in mouse. In the same 2014 paper, we have found that the worm during normal well-fed conditions, the sensory neuron ASI of the hermaphrodite release TGF-beta signaling and affect interneurons in the gonad to promote the increase the synthesis of F-series prostaglandin, which in turn increase the rate of fertilization. However, when the worms are crowded or when the worms are hungry, they will sense the signal in the environment in the form of Dow pheromone that will act and inhibit the ASI to release TGF-beta, and therefore will reduce F-series prostaglandin synthesis and reduce the rate of fertilization. Therefore, we have shown that the environment affects the hermaphrodite fertility. So next we ask, does the environment affect the male fertility too? And that's become the question of my thesis study. Let me rephrase the question in a more tangible way. Does the environment affect sperm motility? So sperm in male are made and stored in the seminal vesicle outlined here. And then during mating, the sperm will use its specialized tails with finger-like structure to sense where the vulva of the hermaphrodite are. And once it's found the vulva, it will inject sperm to the tract through the vulva inside the uterus of the hermaphrodite. Once inside, the round non-motile sperm are activated and become motile, and they target the spermatheca. The males use its sensory neurons to sense the environment. The major group of 12 active sensory neurons are here responsible for many of the sensation ability. And these cell amphisensory neuron are sent dendrite anteriorly toward the nose, and they all terminate in sensory cilia, the structure they use for sensing the environment. So the next question becomes, what would be the olfactory receptor that function in the sensory cilia here that allow it to know the environmental cue? C. elegans genome has around 1,300 homolog of olfactory receptor, and they are called chemosensory 17 receptor. They're all G protein couple receptor with 7 transmembrane domain. Because of the snake shape of this protein class, they're called serpentine receptor. These receptors are further divided into 22 families based on homology. In order for us to figure out which member of which family may be the authentic chemoreceptor in cilia that mediate the sensing of the environment in males, we first must figure out which of them are required in males to promote sperm guidance. In order to address that question, we must be able to assess how efficient the sperm target, the spermatheca. For that, we use sperm guidance assay, where we take wild types or mutant males, and then we stain them with miter tracker, allowing us to see the red sperm. And then we make that stained male to wild type hermaphrodite. The sperm, after transfer into the uterus, become activated and grow a single pseudopod allowed to be motile. And then the sperm will start crawling toward the spermatheca, or zone 3. After 1 hour, we take a snapshot, which looks like this, and we use the percentage of sperm that target zone 3 as the one measure of the sperm targeting efficiency. And wild type sperm target zone 3, about 92% of them get to zone 3 in 1 hour. Using this assay, we screen 10 different serpentine receptor mutant males, and we found out of 10 mutant males, only one of them, SRV13, delta 1 mutant male that produce sperm that target zone 3 would reduce efficiency, only 66% compared to 92%. And this is a representative image of the sperm distribution of the SRV13 deletion 1 mutant sperm. As you can see, many of them did not reach zone 3. The deletion in SRV13 genes take out part of exon 3, and most of exon 4 is a quite big deletion relative to the gene size. In order to confirm ourselves that this gene is important in males to promote sperm guidance, we decided to use homologous recombinations mediated by the removal of transposon to create a second deletion in the gene SRV13. We call it SRV13 delta 2, and we found that the newly created SRV mutant sperm also target zone 3 would reduce efficiency, similar to the previous SRV mutant sperm. So now we're confident, and we conclude that SRV13 function in males to promote sperm guidance. Let's look at SRV13 locus on chromosome 2. So within a 20 kb stretch of DNA, SRV13 is clustered with 6 other SRV genes, SRV1, 2, 3, 5, 12, and 16. SRV4 is a pseudogene. The most closely related to SRV13 is SRV5. Most distantly related is SRV16. So we wonder if any of these other SRV genes may also play a role in male to promote sperm guidance. To address that question, we ordered the next available SRV mutant worm strand, and we have SRV13 deletion mutant. When we made the mutant male to wild type hermaphrodite, we found that the SRV16 mutant sperm also target zone 3 would reduce efficiency 67% compared to wild type, as in 92%. So we conclude that SRV13 also acts in males to promote sperm guidance. Now we wonder whether the loss of function of this SRV GPCR would cause some general sickness that cause sperm guidance defects as a secondary effect. So to test that questions, we decided to perform a reciprocal mating. This time, we'll take wild type males and mate it to mutant SRV hermaphrodite, and we found that the wild type sperm target the zone 3 of the SRV mutant hermaphrodite with high efficiency, the same as wild type. So we conclude that the SRB GPCR do not function in hermaphrodite to promote sperm guidance. The implication of this is that the loss of SRB GPCR do not cause general sickness. Then we look back at the SRB locus. We found the next available SRB mutation, SRB5 deletion. This deletion takes out the splice junction at exon 2, therefore causing a skipping of exon 2, and the rest of the exon have frame shift. And we found that the SRB5 deletion mutant sperm also target zone 3 with reduced efficiency. So we conclude that SRB5 also acts in males to promote sperm guidance. Next to SRB5, there are also SRB2 and 3. So we decide to take SRB2, 3, and 5 both out by homologous recombinations to see whether we'll see an increase in the severity of defects or just the same. And we found that the loss of SRB2, 3, and 5 mutation in sperm causes sperm to target zone 3 even with further reduced efficiency. 61%, which is statistically lower than 69%. So we conclude that SRB 2 or 3 may also play a role in male to promote sperm guidance. We know that SRB 15 and SRB 16 both function in male to promote sperm guidance. We wonder if they function in the same genetic pathway or if they function in parallel pathway. If they function in the same genetic pathway, then we would expect that the loss of both of them will have the same level of phenotype as the loss of any individual of them. But if they function in parallel, then loss of both of them will cause a more severe phenotype. And we use homologous recombination to take out SRB 15 and 16. Unfortunately, SRB 12 happened to be right between, so we also take out SRB 12. We found that the triple loss of the 3 SRB gene, 12, 15, and 16, caused the sperm to be even target zone 3 even more deficient. So only 49% the target zone 3, which is much lower than the SRB single mutant sperm, which target zone 3 at 66 or 67%. So we conclude that the evidence is suggestive but not yet conclusive at this point that SRB 13 and SRB 16 function in parallel. However, later on in the presentation, we will provide 2 more lines of evidence supporting this hypothesis that SRB 13 and 16 work in parallel pathway. SRB are G protein coupled receptor. Therefore, we expect their downstream signal transducer to be the heterotrimeric G proteins. And the C. elegans genome has 1 paralog of G alpha S, 1 paralog of G alpha Q, and 1 paralog of G alpha olfactory. Unfortunately, the loss of function of G alpha S and G alpha or G alpha Qs cause embryonic lethality. So for the sake of easiness, we work with the gain of function mutation for these 2 genes. We found that the gain of function of the G alpha S does not affect sperm distribution to zone 3 as a wild type level here. However, the gain of function in G alpha Q causes a strong reduction in the sperm distribution to zone 3. So we conclude that G alpha Q echo 30 play a role in males to promote sperm guidance. But more importantly, the loss of function of G alpha olfactory, GOA1, cause a strong sperm guidance distribution sperm in zone 3, 53%, which is similar in the severity to the most severe SRB mutant sperm, the SRB 13, 12, and 16 triple mutant sperm. So we conclude that G alpha olfactory GOA1 is a likely downstream component gene that function in males to promote sperm guidance. So in summary, we have identified a SRB GPCR network that function in male to promote sperm guidance. Particularly, we found that SIV 13, 16, and 5 function in males to promote sperm guidance. SIV 2 or 3 may also play a role. We also found that SIV 13 and 16 do not function in hermaphrodite to promote sperm guidance. The indication is the loss of the SIV GPCR do not cause general sickness. Also, we found suggested evidence that SIV GPCR may function in two parallel pathways. And we also identify a downstream G-alpha olfactory GOA1 that function in male to promote sperm guidance. So what's wrong with the SIV GPCR sperm? So we take time-lapse images of this SIV mutant sperm in Y-type hermaphrodite uterus. And we use that to calculate average velocity and reversal frequency. We first found that all of the SIV mutant sperm are moral. So that means they have all been activated. And then the second thing we found is that the SIV mutant sperm are either slow, as in SIV 16 mutant sperm or SIV 5 mutant sperm, or the SIV mutant sperm is confused, like the SIV 13 mutant sperm. Also, interestingly, the loss of SIV 13, 12, and 16 results in abnormal sperm motility. This is a combination of the most severe phenotype of the SIV 13 and SIV 16. Together, this is the second line of evidence supporting the hypothesis that SIV 13 and SIV 16 function in parallel. So now we have identified SIV genes that function in males to affect sperm motility. So the most simple explanation is that the SIV are expressed in sperm. So we decide to address this question by a quantitative PCR approach. We use SPE9 and SPE11 genes, which are known to function and express in sperm as positive control for the genes that are enriched during spermatogenesis. We indeed found a very strong enrichment of the messenger RNA of SPE9 and SPE11 in worms strains that produce a lot of sperm compared to the worm strains that produce no sperm at all. With this good positive control working well, we, however, found that none of the SIV gene that we look at are enriched at the transcription level during spermatogenesis. We decided to test it in a more direct approach. We decided to look and see if the SIV13 and SIV16 gene are expressed endogenously in sperm. To do this, we use a TD tomato knock-in approach where we use homologous recombination to insert TD tomato into right before the stop codon of SIV13 or SIV16. We also insert UNC119 rescue fragment as a positive selection marker. In order to control for our ability to observe the TD tomato signal in sperm, we also make SPE9 TD tomato knock-in too for that. We are very fortunate that the TD tomato insertion do not affect the gene functions. Particularly, the SIV13 and SIV16 TD tomato knock-in mutant worm produce mutant sperm that target the zone 3 of Y-type hermaphrodites with Y-type efficiency, suggesting that the insertion of the TD tomato did not abolish the function of SIV13 or SIV16. Similarly, we would expect the SPE9 loss of function worm to be sterile because SPE9 is required in sperm for fertilization. However, SPE9 TD tomato knock-in worm are fertile. We believe that the TD tomato insertion did not affect the gene function of SPE9 either. With that all clear, now we look at the endogenous expression of this gene in sperm. So we found SB9 TD tomato signal in sperm as a positive control here. So the sperm are inside this yellow outline, and the outsides are the sperm residual body. However, we failed to find SRB13 or SRB16 endogenous expression in sperm of either hermaphrodite or in sperm of males. So we failed to see their expression in sperm. We conclude that SRB GPCR are not expressed in sperm. In summary, this is summary about sperm. So we have shown that the SRB mutant sperm are moral, and they have all been activated. Therefore, their problem happened downstream. We also found that the SRB mutant sperm have abnormal motility. They are either slow, as in the SRB13, 16, or SRB5, or the mutant sperm are confused, as in the SRB13 mutant sperm. We also show that SRB G protein couple receptors are not expressed in sperm. Therefore, we believe that they function in a non-cell autonomous manner. They function somewhere else. To figure out where they function, we use the predicted promoter of SRB13, and we drive expression of GFP in male. We found that this predicted promoter drive expression in 6 cell bodies, and when we overlay this image with a map of all the sensory neurons that has been labeled with DID, a pansensory neuronal marker, to identify what cell is what, we found that the predicted SRB13 promoter drive expression exclusively in 3 pairs of sensory neurons. They are the ASI and ASK, which stand for amphid single ciliated sensory neuron I and K, and AWB, which stand for amphid winged sensory neuron B. The ASI-ASK has a single linear cilia. The AWB have a single cilia, however, it's branched, so it's branched more, like 2 or 3 times. However, what we've seen was not the endogenous expression of SRB13. To look for that, we look at the TD tomato signal in the SRB13 TD tomato not in mutant males. And we found that SRB13 is endogenously expressed in 2 structures that appear like a sensory cilia. This looks like the basal body, and this looks like the axoneme of the sensory cilia. Again, this are linear, and that doesn't look branched. Also, we observed SRB13 endogenous expression in several puncta in this region that look around the same region as the cell body of the sensory cilia. So we are guessing that these are the trafficking population of SRB13 genes. However, in order to test whether the cilia-like structure are truly cilia, so we take the TD tomato not in one, and we express a pansensory cilia marker, driven with OSM6 promoter. And here you see is the sensory cilia, and you can clearly see the co-localization between the sensory cilia and the endogenous expression of SRB13. Therefore, we're confident that SRB13 is expressed in the sensory cilia, most likely belong to ASI and ASK sensory neuron, but not AWB, because that one tends to have a branched cilia. Similarly, we drive the predicted promoter of SRB16. We drive GFP in males, and we found a broader expression pattern. So we see expression in pharyngeal muscle cell, PM4, PM5, and also in 4 pairs of the sensory neuron, ASI, ASK, ASH, and AWB. And then we look at the endogenous expression of SRB16 of the TD tomato signal of the SRB16 TD tomato not in mutant males. We found that SRB16 is expressed endogenously in a pair of interneurons called I1 interneurons, also in pharyngeal muscles PM4 and PM5, and also in some other unidentified neurons and also in the 4 sensory neuron cell bodies of the ASH, ASI, ASK, and AWB. Next, we asked if the SAB16 is also expressed in the cilia as in the SAB13 case. However, when we drive expression of the pan-sensory cilia marker, we failed to see a co-localization between SAB16 endogenous expression and the sensory cilia. Therefore, we conclude that SAB16 is not expressed in cilia. However, expression doesn't mean that's the place where it functions. Therefore, we ask whether the SAB GPCR functions in neurons to promote sperm guidance. For this, we ectopically express SAB13 under the pan-neuronal promoter, UNC119 promoter, and we found that in 3 independent lines, the ectopic expression of SAB13 under pan-neuronal promoter rescue the sperm distribution of the defect here of the SAB13 mutant sperm. Similarly, we found that ectopically expressed SAB16 under pan-neuronal promoter UNC119 also rescue the sperm distribution defect to zone 3 to wild type level for the SAB13 loss of function mutant. We decide to dig a little further for the SAB13 and ask whether SAB13 function in a smaller population of the neuron, the sensory neuron, to rescue the SAB13 loss of function mutations. And we found that in 2 independent lines, the ectopic expression of SAB13 under a pan-sensory neuron promoter, OSM6, also rescue the sperm distribution defects to wild type level. And this last one is a control promoter where we drive SAB13 under a body wall muscle promoter, MYO3 promoter, and this one failed to rescue the sperm distribution defect. Therefore, we conclude that SAB13 function in sensory neurons to promote sperm guidance. In summary, we have shown that the predicted SAB13 promoter drive expressions in male sensory neuron, ASI, ASK, and AWB. And we also show that SAB13 is expressed endogenously in cilia of the male ASI and or ASK sensory neurons. We also show that SAB13 functions in male sensory neurons. Similarly, we shown that the predicted SAB16 promoter drive expression in males ASI, ASK, ASH, and AWB sensory neuron. And that SAB16 is expressed endogenously in the cell bodies, but not the cilia of ASH, ASI, ASK, and AWB. And that SAB16 functions in male neurons. Up to this point, however, we do not know what is the molecular mechanisms that somehow could explain some genes, some G protein coupled receptor genes that function in sensory neuron that could affect sperm motility at a distance. In our 2014 paper, we have found that the sensory neuron of hermaphrodites release TGF-beta and affect the transcriptions program of the oocytes, and in turn that promote the F-series prostaglandin synthesis. So we wonder if the sensory neurons of males, which are acted through the GPCR, would also affect the transcription of the sperm or sperm precursor. So to address this question, we compare the transcriptomes of the males of the SAB mutants compared to control males. For this, we choose 2 SAB mutant males, SAB13 single mutant males, and SAB13, 12, and 16 single mutant males. The rationale is that whatever genes that alter by both SAB mutant males, are most likely be regulated by SIB13. And whatever genes that alter only in SIB13, 12, and 16 mutant males, but not in SIB13 single mutant males, those genes will most likely be regulated by the parallel SIB12 and 16 pathway. So using that approach, we found that roughly 1,000 genes are upregulated in either SIB mutant males, and roughly half of that, 500 genes, are downregulated in either mutant males. In order to know which of the genes here are spermatogenics or involved in the process of making sperm, we filter this list through a top 1,000 gene transcribed in sperm, and we found roughly about 80 genes, 80 spermatogenic genes upregulated in either SIB mutant males, and about half of that downregulate in either SIB mutant males. When we further characterize these genes, we found two interesting trends. The first trend I want to point to are the sperm structural components genes. So we found that out of the 65 genes that only upregulated in SIB 13, 12, and 16, 16 of them code for sperm structural components. So we conclude that normally SIB 16 or 12 suppress the expression of the sperm structural genes. The second trend I would like to point to are the mitochondrial component coding genes. So 8 out of 19 genes that downregulated in both SIB mutant genes are the one that code for mitochondrial components. Therefore, we conclude that normally SIB 13 would promote sperm metabolic genes. So all of these lead us to the following working model, in which the SIB 16 GPCR function in the cell bodies of ASH, ASI, ASK, and or AWB, and it acts through the downstream G-alpha olfactory GOA1, and then signal through an unidentified neuroendocrine mechanism that suppress the expression of sperm structural genes. And on the other hand, SIB 13 functions in the tip of the cilia of ASI and or ASK sensory neuron, and function through a downstream G-alpha olfactory GOA1, and then act through an unidentified neuroendocrine mechanism to promote sperm metabolic genes. I want to one important implications in this study in C. elegans is that normally we think that the males would maximize its potential to lead us to pass the genetics to the next level. The males would try to make copious amount of sperm that highly efficient all the time as long as they can, right? However, in here we've shown that in C. elegans males, they regulate their reproductive output based on the environment. So with that, I want to thank Michael and our lab for being very, very great, make me very happy, so I want to first and foremost thank Michael. He is the home provider, and especially for us, international student and international scholar who left our home in our home country and come here. He provide home-like environment. That during the early year of my graduate career, I really hate holidays because then my family would go away to their family and I will have no more family. So I really hate Christmas. Until I have a family now, I don't hate Christmas anymore. But anyway, so this Michael provides a really amazing environment that's full of nutrients and allow me to soak up all of the nutrients and grow. Also, he provide fertilizer to the modern games, so that can refresh our mind and make us work more intuitively. Also, I have learned a lot, not only from Michael. Actually, I only learned little from Michael. I hope to learn more from Michael. He is very smart. I want to learn more. Anyway, so I have learned a lot from other lab members like Sejin and Saming, who are great teachers, who taught me a lot, many things and cloning, and also many things about life. This is Catherine, Dr. McKnight, Saming Dr. Hoang, Sejin is Dr. Lee, and Jack, he will soon be Dr. Weber. He is a third-year medical student now. I'm rooting for him. This is Wes, Dr. Edmonds. He taught me a lot on some guidance and also on extracting lipids from worms. It's earned me many great success, so thank you very much, Wes. I would like to thank Pauline, who is not here. So this Dr. Kadi, she is now she's an embryologist in New Zealand now, I believe. And also, I especially thank Jess. She is a very amazing, supportive lab mate. She is the most persevered graduate student that I know. She is very hardworking. I believe that she will have a great career after here, and she'll be a very happy person. Thank you, Jess. I would like to especially thank teams, our lab mate, lab manager, and also our lab engineer. He is one of the smartest guys I know. He devised many contraptions, some for me. Very smart guy. Sometimes I think that his height has to do something with his intelligence. You see, he has more height than me. Maybe that's why. I would like to thank my committee, Michael, Brad, Jim, Chenbei, Ken, and Dr. John Wang. They're all very supportive and not only during meetings, but also call for advices outside of the meeting context. I'm very grateful. I thank our CMDB teams, which are very supportive. Thank Brad, Alicia, and Nan, and also CMDB friends and colleagues. I thank them very much for even skip some class and come here to be with us. Very grateful for the strong support. I love the sense of community that we have having now. I hope that we will be a strong community. I thank the CGIB staff that very supported in many ways. I especially thank Jiliang Tin and Matthew Such for training me a lot for this presentation. So they asked many questions and many suggestions. Thank you very much. I thank the funding agencies, the NIH, the Muscular Dystrophy Associations, and also CMDB and CDIP for not only paying for my salary, the reagents that I use in the lab, but also pay for the travel expenses, and also for many great free meals and great free t-shirts and many other things. Also thank the Sierra Genetic Community Center and the Japanese Knockout Consortium for providing this valuable one strand that saved us a lot of time. We built on something already there. Also thank One Base, One Atlas, and One Book for packaging knowledge into a very fast, almost like fast food ready that I can absorb very quickly. And last but not least, I'd like to thank my family, especially my father and my mother, who could not be here. My father taught me a lot about all the values I have now, how to be a good person in the society, how to how to contribute, how to contribute. Anyway, I'm sorry. To be a good person and contribute and work hard and be happy. Also, I thank my wife, a wonderful person who helped me, support me a lot. One interesting thing I want to point out that my wife and her family are from the north of Vietnam. My family are from the south of Vietnam. Without this 2 doctoral training at UAB, we would not meet each other and have a wonderful baby, Halvey. So we deeply thank the UAB doctoral programs. And we hope that our baby will grow up, become a scientist like us, not a professional singer. With that, I would like to take questions. And thank you everyone for being here. Yes?
So if I remember correctly, if like mathematics, if they become starved or stressed, they stop contributing as much nutrients and whatnot to the yolks of the egg to help maintain for survival.
Yes.
It sounds like the same thing could be happening with males, where they're basically down regulating the metabolism of their sperm as a way to try to conserve nutrition for themselves. If you starved male worms prior to doing your mitochondrial tracking or your sperm tracking assay, do you think starved males would also have a sperm tracking speed?
So Michael also thought of that, and he had tried some preliminary experiment. He starved some males 24 hours prior to mating. However, they seem to mate fine, so we don't know.
The answer was no difference.
No difference.
Yeah, they targeted, they didn't care. They don't care about, males don't care about food. Females do, but males don't.
Also, I'm very grateful to Scott and Don who can me a lot with the confocal microscopy. They are very, very enthusiastic, very helpful. Also, they provide us with the ID and specific instruction on how to scan the sensory neurons. They've been great friends and very, very, very helpful, very skillful also. Great picture. Yes.
So you and your arrays, or maybe it was gene RNA-seq, in the SRV13 you only had 1 gene that went up. It seems like easy targets, right?
Yes, I mean.
On your data set that you had for SRV13, you said only 1 gene went up.
However, we do not know what this means. Like, if a gene is regulated about SRV13, then we would expect that they would be in this category. Like, they should both be affected. It should affect it in both SRV mutant males. We don't know what it means for some gene to be affected by only SRV13 mutant males.
What I was referring to is, can't you just knock out that gene and see if it gives you the same phenotype?
Yes, that is something that we will try next. For now, for this current manuscript, we are thinking of finding big trains, and then for future study, we will look specifically at candidates. Since we have a small number of candidates, we can test them out one by one.
Can you transplant, I guess, primordial germ cells into a population? So your notion is that this is autonomous of the sperm, right?
Yes.
And so can you take sperm cells from wild type animals, put it into this animal that's SRV13 null, and ask if they fertilize fine, they go motility, and everything's good?
That is possible. So we haven't done that yet. Technically,
I
it's possible. We can
neuron
do.
rescue. I realize that. I'm just trying to do the reverse thing.
Yeah, we haven't done that. Yes.
So what do you think is actually signaling these GPCRs? What is activating them?
Okay. So because so the answer is we don't know what are the signals that activate SLB13, but our best guess are the pheromones. The reason is because ASI, in this case, sensory neuron, have previously been indicated in signaling pheromones. So therefore, we're thinking that SLB13 may be mediated in some other form of pheromones, maybe male-derived pheromone or something like that. We don't know. Yes?
Have you checked the morphology or anything of the neurons in the mutants to see if they look the way they should, if that's where SRB is normally located?
We have not looked at the morphology.
Or
No, we
functionality,
haven't.
I don't know.
We have not done that yet.
So you localize some of these things in the cilia, right? So do you know if the males that have cilia defects, does the sperm totally alter this?
We also attempt. So the question is, would a male have cilia defect? Would those mutant males also have trouble with getting their sperm to zone 3? So we were in the process of addressing that question. We obtained a mutations in, I believe it's diff5. And then, however, when we make the mutant males of diff5, that mutant mate very inefficiently with hermaphrodites. I tried many matings. Only 1 or 2 hermaphrodites mated. So with a very low number, we don't know. At this point, we will try again to get higher number. We don't know. But based on the current result of 4 gonad size, then we have 3 gonads that target well, 1 gonad that does not target well. So we don't know. We would like to know that. We would try again. Yes.
You've mapped out a bunch of GPCRs, and you see with some of them just small incremental steps in changes. Did you see any other phenotypes, any other differences in these animals with when you've knocked out 4 or 5 of these GPCRs?
We have not looked carefully. The answer is no. We have not found any other phenotype. Michael has looked at the sperm size. The sperm size look about the same in either SLB13 single mutant or SLB13, 12, and 16 single triple mutant. Compared to wild types, the sperm size are the same. The morphology of the males about the same. And also very recently, Michael done some lifespan study, and he found that the SLB13 triple mutant males maybe have longer lifespan than the other 2 in the SLB13 mutant males and wild type, but we need more number for that one. Sorry. Since you asked that, I would also like to show you this hidden slide. Actually, the SLB13 not only affecting the gene affecting the sperm, it also affects multiple processes. So, for example, we found that many collagen genes are down-regulated in both SLB13 mutant and SLB triple mutants. And also other immune response genes are affected, and metabolism gene also affected.
So you look at the male sperm. What about the hermaphrodite sperm? Does that have defects in the chemotaxis or transport versus sperm?
We have not looked at the hermaphrodite sperm, but the hermaphrodite mutant of the SLB genes seems to look wild type and also have baby normally. We don't see a drastic reduction in growth size. Maybe no more than 3 or 4 folds, or 2 folds or 3 folds. They seem to grow with normal rate. We have been missing something. We don't know yet.